Get going. So I'd like to welcome everybody to the first of our events in what we are calling Rare Disease Week. People will know that Rare Disease Day is annually now on the 28th of February, or where there is the 29th of February, it occurs on the 29th of February. And we have a number of events lined up for this week. To celebrate Rare Disease Day. And so this is the first of our events, but please do look for the others because they involve parents of children who have rare diseases. So we have an event tomorrow, Tuesday, another on Wednesday, and another on Friday. And then those of you that are St. George's people, please come and join our grand round on Thursday and come to our stall, which will be in the canteen as well. So please do look out for those events. I should have introduced myself. I'm Frances Elmsley. I'm the Clinical Director for the South East Genomic Medicine Service Alliance. But for today's event, we're really, really grateful to Richard Thompson, who is the Professor of Molecular Hepatology at King's College Hospital and King's College, University of London. Richard is going to talk to us about the genetics of cholestasis. He has a long, oh, my mic is very echoey, cannot hear me properly. Sorry, apologies for that. I'm not sure there's very much I can do about that. It may be something about the setup. Hope the same does not apply to Richard. Richard has a huge amount of experience and has an extensive research background in identification of genes that cause cholestasis. So without more ado, I'm going to hand over to you, Richard. Thank you very much.
Thank you, Frances. I'm delighted and honoured to be the first event in Rare Disease Week in the Southeast. And I indeed have been looking for and at genes involved in cholestasis for 3 decades now. And I want to start by giving some credit to the myriad of people who have contributed to the things I'm going to say today. And highlight 2 people in particular. I'm going to embarrass them: Sandra Stroutlix, who has been working with me for the whole of those 30 years, and Sammy Aluni, who is the current lab manager at King's, running the multimodal service, I think it's called, and in particular the liver molecular genetics service, much of the data of which I'm going to present today. I do have some conflicts. I'm not going to talk about them in detail. In fact, I'm not going to mention any of these drugs, but apart from some published, very brief published data today. So there's a few concepts that I want to try and get across today, and they relate to our understanding of genetics and the utility of genetics, but also specific relevance to the diseases in which I'm interested. So when we started off using genetics as a tool to investigate some of these diseases, what we did was we got groups of patients who were highly phenotypically similar. They were generally cohorts of patients that had early onset severe disease, and we used those to identify genes. And we identified similar patients using clinical history, immunohistochemistry, and we sequenced those, and sure enough, they had similar genetics and they had similar phenotypes. And the consequence of this, and this went on for several years, is that we've got numerous publications covering the initial presentation and the clinical natural history of these diseases. And in some ways, we've defined diseases based on the strong correlation with the genetics. And this is obviously now in the literature, published literature, in textbooks and things like that. And we've got these clearly well-defined diseases. However, since then, we've switched and we're now using large panels or exomes or genomes. We're testing patients who've got quite diverse phenotypes. They're nothing like, in many cases, the original patients that we used to identify many of these genes. So they really haven't got the disease as it was published. We're also, to make life more complicated, we're finding patients with heterozygous variants, obviously with phenotypes, that's why they've been tested, in genes that otherwise were thought to behave as recessive genes. We're finding variants which are not complete loss of function variants. We're finding variants which have got retained function. And we're finding, obviously, like everyone else, large numbers of variants of uncertain significance, which is the bane of most people's lives these days. I believe what we're trying to do now is understand what is causing the patient's phenotype. And from that, we want to predict what the likely natural history of the disease is for that individual, both for the family's sake and our own. Use that information the best we can to identify the best form of treatment and also predict likely complications and likely outcomes of those treatments. So I'm not really, in my head, in the business of trying to make diagnoses. I really want to understand what's happening to patients. And to be able to do that, we need to really understand what the gene and the consequent protein does. We need to understand what a loss of function might do. We need to understand what a partial loss of function might do. And in that context, we need to understand, is there a threshold? Or is there a much more complex relationship between the amount of residual function and any phenotype? And then we get bogged down in all these really confusing terms of predispositions and modifiers and reduced penetrance and risk alleles. And these terms have arisen over the last few decades because it is clear that not all diseases have got exactly one-to-one relationship with severe early onset disease and complete loss of function genetics. So these are really confusing terms, which I hate. But the truth is that those terms have arisen because we're trying to cram everything into a recessive or dominant model, which doesn't always apply. So in my dreams, eventually we'll get rid of all of those terms because they really are models which are sometimes useful, but actually end up oversimplifying the situation. And my mantra is that we want to use genetics and find variants which we believe are contributing to the phenotype. Now, using a recessive or dominant model, we can try and sort of visualize the relationship between protein function and phenotype. And this might be the whole disease phenotype. It may be particular aspects of the disease, but you can apply it to whatever phenotype you like. And conceptually, if something is a recessive, the relationship looks something like the one at the top, where we all hope we're up at the top right-hand corner, where we've got full function and we're phenotype free. The original patients, which I've just sort of alluded to, are the ones in the bottom left-hand corner, where they've got neither allele functioning at all, and they've got the severe early onset disease. And if something is behaving like a recessive, then by definition, where the blue line is at 50%, one copy working, one copy not working, they are also phenotype free. I believe for most diseases or genes that behave like recessives, we don't really yet know what the relationship looks like. All we know is that the heterozygotes are disease free. We don't really know whether you need 40% to be disease free or whether 10% is enough. Obviously, some genes have been extensively investigated, others less so. And in the case of so-called dominance, if it's a haploinsufficiency dominant, then we believe that 50% is enough to cause disease, 50% loss, and that somewhere between 50 and 100 will be the threshold between being disease-free or not. And so you've got similar curves like that. Obviously, other forms of dominant disease exist, but that's just a sort of haploinsufficiency version. But today I'm going to talk about diseases that are originally described certainly as recessives because we identified patients down at the bottom left-hand corner. Then, with that little bit of genetic background, I want to move to a bit of liver physiology. And the liver is a large organ. Of course, it's by far the most important organ in the body, but that's just my bias. It has got an unusual blood supply, as I hope most of you know, in the sense that the majority of the blood going to the liver comes from a previous capillary bed. So it's a venous drainage of the intestines, the spleen, and the pancreas, all drained via the portal vein and then flow through the sinusoids, which is a very low resistance system, into the hepatic vein, from where they go straight back to the heart. However, in the liver parenchyma here, flowing in the opposite direction to that blood is bile. And bile, of course, is made by the liver and ultimately flows into the intestine, where it contributes to absorption of fat and fat-soluble vitamins. So, but bile is a mixture of excretory products and products which you want to help us absorb dietary products. And bile's formed in canaliculi, which are space, which I've tried to show with this arrow, the space between adjacent hepatocytes. And although hepatocytes just seem to be one great big mass of cells, they are actually epithelia. And the basolateral surface is exposed to blood and the apical to bile. If we zoom in on a canaliculus here, it obviously doesn't look like this, but this is a cartoon demonstrating the important functions of a canaliculus. We've got transporters in the membrane. I'm showing two adjacent hepatocytes here, forming a canalicular space, which is the very distal branch of the biliary tree where bile is formed. And we'll go through some of these transporters in a second, but just to show that obviously to have any useful function, the canaliculus is sealed at the top and bottom there by tight junctions made up of claudins and associated tight junction proteins, so-called TJP2s, which are actually cytoplasmic proteins associated with those tight junctions, which I've shown there and we'll come back to later. If we zoom in on this membrane where bile's formed, these are the key players. So we've got an hepatocyte on the left-hand side, we've got the canaliculus where bile is on the right-hand side. In everyday liver medicine, the most obvious feature that brings most of our patients to our attention is that they have jaundice, and they usually have conjugated jaundice because the bilirubin has been modified in the liver to make it conjugated, but then there's a failure of transport, and MRP2 is that transporter. Mutations in MRP2 are extremely rare but cause Dubin-Johnson syndrome. I'm not going to talk about that today. The major transporter of bile acids, which are the detergent in bile, are BSEP, and BSEP is the bile salt export pump. And we identified the gene for BSEP approaching 30 years ago. And it is the main driving force of bile by transporting bile acids. Now, if we just transported bile acids and they were left alone in bile like that, they would cause massive destruction of the biliary tree because they are detergents and are in, like every cell in our body, the biliary tree is composed of lipid membranes. And so we cannot have high concentrations of highly detergent bile acids in bile. So to get around this, what we do is we package those bile acids in mixed micelles, which are about 2/3 bile acids and 1/3 lipids, mainly phosphatidylcholine. And to get phosphatidylcholine available for transport into and packaging into micelles, MDR3 translocates phosphatidylcholine from the inner leaflet to the outer leaflet, where it can be extracted by bile acids and packaged into mixed micelles. FIC1 is another phospholipid transporter at the bottom, which I'm not going to talk about today, but that's an aminophospholipid transporter that transports lipids in the other direction. It internalizes them. And I'm not going to talk about ABG5G8, which is the cholesterol transporter at the top. So if we decide that is the normal physiology, what goes wrong? So the simplest to understand is indeed BSEP deficiency. If you've got a complete absence of BSEP, you've got retention of high concentrations of bile acids in the liver, you've got very low concentrations of bile acids in bile. So the liver is damaged by these very inflammatory detergent bile acids in the hepatocytes, so they have a hepatitic presentation, often with jaundice at presentation because of the secondary liver damage. Sometimes they clear their jaundice before it recurs. So they've got severe liver injury, but they've also got severe malabsorption because they've got low concentrations of bile acids in the bile and subsequently in the intestine where we actually want them. So that was the original disease, the early onset severe disease, complete loss of function of BSEP, very rapidly progressive disease, very high incidence of liver cancer, and the majority become to transplant in the first few years of life. However, what happens if you've got reduced BSEP function? And we've now got patients with all sorts of levels of BSEP residual function, and we need to understand what happens to them. Well, I think the truth is that at any one moment in time, you either have enough or you don't. If you've got just enough and you don't have retention of bile acids, then you don't have cholestasis and you have a happy liver. But if you just haven't got quite enough to transport bile acids at that moment in time, then you will have retention of bile acids and you will have cholestasis, even if it's only on a temporary basis. So I believe there is a threshold. The threshold, exact threshold, may vary depending on the demand, and depending on whether you've been exposed to other drugs or whether you're pregnant, etc. But I think the relationship overall between being disease-free and the amount of BSEP function does indeed look very similar to the curve I'm showing you now. And that generally patients with 50% of ideal function are fine. I think that if you go down to around less than 20% function, you've got permanent cholestasis. But we've got patients who get episodic cholestasis, they get drug-induced cholestasis, they get cholestasis of pregnancy. Now, why is that happening? Why are we getting patients who have got intermittent disease? Well, if you look on Nomad, and I'm sure most people listening to this are very familiar with Nomad, and we look at this gene, we'll find there's this variant here, which is called p-val444-ALA. And if you look at the allele frequencies on here, they're somewhat peculiar, aren't they? Because the minor allele frequency is well over 50%. This has come about because when we originally published the sequence of BSEP, we actually said that the valine was the alanine was the wild type, the alanine, which of course is the slightly more frequent. But what's become clear is that actually the alanine has got significantly reduced function compared to the valine. So now the valine, even though it is the rarer of the two alleles, is now considered to be the wild type. So this valine 444 alanine actually, in some people, it actually reduces transport by about 50%. So homozygotes only have about 50% of transport. They, of course, are disease-free. In fact, homozygotes are a third of the population. So this variant on its own, even homozygotes, usually do not manifest disease, although statistically, alanine homozygotes compared to valine homozygotes have got 3 times the risk of cholestasis of pregnancy or drug-induced cholestasis. But overall, 83% of people have got at least one copy. So we do report this variant because it is functionally important, but obviously it is nowhere near making a diagnosis on its own. I think when it becomes clinically important, though, is where we've got patients who've got one non-functional allele and they've got the V444A on the other copy, because they will now be down to about 25% of function. So these are the people who get drug-induced cholestasis and cholestasis of pregnancy because they're hovering right on the brink of having enough transport. And most of the time, they are fine. They're not cholestatic, but it only does take something to come along and either reduce the function or increase the requirement for function, and they retain bile acids and become symptomatic. So these are the people sitting on this threshold here. Now, if we go back to people with biallelic disease, people with early onset disease, and we start looking at their genetics in more detail, we can start subdividing them depending on what actual mutations they have. So this is exclusively patients with biallelic disease. Both copies are mutated. And we did a very simple split here, and we just said there's these 2 variants which are relatively common in Europe, E297G and D482G are the 2 most common European variants, and they did seem to be behaving slightly differently, and indeed in vitro have got some retained function. Now, if you've got one copy of those in combination with anything else, whether it's a truncated one or a missense one, you're in the BSEP1 group in this particular analysis, and that puts you, as you can see, with a native liver survival, in other words, with transplant-free survival of over 50% by the time you reach adulthood. The other extreme is what's called BSEP3 in this particular publication, These are patients with 2 protein truncating mutations. So these are more like the very original patients that we described from the Middle East. And they've got no residual function in vitro and have got 2 truncating mutations. As you can see, although they survived for a few years, none of those patients made it into adulthood with their own liver. Unfortunately, the BSEP2 is the group in between, so they just don't fit either of those other 2 groups. They are the biggest group, and at the moment we're trying to dissect the BSEP2 category into a slightly more refined way so that we can give a clearer indication. But at the moment, there are a ragbag of patients who undoubtedly some have got far more retained function than the so-called the BSEP1 group, and there's others that have got as little function as the BSEP3 group. So this is just, regardless of treatment, this is just overall native liver survival in a historic cohort from around the world based on their very simple dissection of genotype. Now, another bit of physiology is to point out that bile acids, which at the top I've shown being transported by BSEP into bile, go into the intestine, but then recirculate via absorption in the distal ileum. And I'm showed an enterocyte at the bottom here, where they are taken up by ASBT, the sodium-dependent bile acid transporter. They're transported out of the enterocytes and back to the liver, where they're taken up by the basolateral transporter, NTCP. So this is a very efficient recirculation of bile acids, which is what happens in normal physiology. We'll come back to ASBT in the intestine in a couple of minutes. But we have historically interrupted that recirculation of bile acids to try and modify the disease. So what's happening in my little cartoon on the left-hand side has become even more cartoon-like on the right-hand side, where I'm just showing transport across the liver at the top and transport across enterocytes at the bottom, with bile in the intestines on the right-hand side and portal blood on the left-hand side. And in normal patients, of course, virtually all the bile acids at any one moment in time are actually on the right-hand side. They're in bile and intestine. They don't hang around inside enterocytes or hepatocytes or in portal blood. Now, patients with low gamma GT cholestasis, like BSEP deficiency, have got a problem with transport of bile acids out of the hepatocyte, and I've tried to represent that by these 2 red circles, and they've got retention of bile salts in the liver, which I've shown by the larger blue triangle. The consequence of that retention is that we get spillover of bile acids into the peripheral blood, and that's, of course, where we measure them in clinical practice. When we measure bile acids in the blood, we're not measuring them in this enterohepatic circulation at all, we're measuring them in peripheral blood. So this is how we can monitor the disease or monitor treatment, is by measuring the overflow from the enterohepatic circulation. Now, what we have done historically is we've got our pediatric surgical colleagues to put a loop of jejunum between the gallbladder and the anterior abdominal wall. And that diverts some of the bile, that's why it's called partial, some of the bile out of the body into a stoma every day to reduce the amount of recirculating bile acids, to try and reduce the amount of bile acids coming back to the liver and therefore reduce the requirement for the liver to transport bile acids, in the hope that that would improve the symptoms and the underlying liver disease. And it works. We've now done this pharmacologically, and these are the 2 papers which we've recently published using 2 different drugs with exactly the same mechanism of action, and they block that sodium-dependent bile acid transporter in the terminal ileum. And so they achieve a non-surgical treatment of what we've been doing historically to reduce the amount of recirculating bile acids. So what we hope we've done is we've transformed a patient whose cartoon looks like the left-hand side, where they've got reduced transport and accumulation with spillover into the blood, to a patient on the right-hand side who's now got a new arrow on the right-hand side, which is either the bile diverted into the stoma in the surgical case, or bile acids diverted into stool in the pharmacological case. But as if we achieve what we're hoping to do, then it means that the recirculating bile acids are that much less. There's no accumulation of bile acids in the liver. And although we haven't increased the transport, we have reduced the retention of bile acids and consequently the spillover into the peripheral circulation, where we can measure it. And in both cases, it works. So what I believe we've done is we've got these patients with more than 25% function. They're the normal individuals. We've got these patients with less than 20% function, and these are the patients that have classical BSEP deficiency. By depleting them of bile acids, whether it's surgically or pharmacologically, what we do is we move the curve to the left-hand side. So these patients with less than 20% and more than 10% approximately are the ones who've responded very well to these drugs. And I got one patient who's now been on one of these drugs for 10 years and has completely normal bile acids, normal liver physiology, normal ultrasound of the liver, which is in stark contrast to her sister who was transplanted successfully a few years prior to these drugs becoming available. Her sister, of course, requiring lifelong immunosuppression. So we've about 50% approximately of patients have responded very well to these drugs, but obviously there's patients still who are on the left-hand side of that curve, and we either need to increase the function or we need to come up with some completely alternative route to improve their phenotype. So that's work in progress. But I think physiologically what we've done, and I think it's useful, to me at least, to try and explain these relationships between function and phenotype status using these curves and understand how the curve moves when we intervene with various forms of treatment. Now, we'll go back to the beginning and look at this illustration of normal physiology, and we'll look at another problem. So in this case, what happens if MDR3 is not working? Well, I've alluded to this already because I've told you that if you've got high concentrations of free bile acids not packaged in mixed micelles in the bile, you're going to get extensive injury to the biliary tree. And so the patients, again, which were originally described with MDR3 deficiency, were early onset severe disease presenting not always in the first year, but certainly in the first couple of years with progressive liver disease. And in truth, this wasn't a form of intrahepatic cholestasis. This was a form of cholangiopathy, or bile duct injury. They became cholestatic, they became jaundiced, they became pruritic later on, but that's only a secondary to the bile duct injury causing secondary liver dysfunction. These patients require early transplantation and respond well to that treatment. However, what happens if we've got partial MDR3 function? Well, I believe what happens if we've got, say, 50% of MDR3 function, we have something looking a bit more like the scenario I'm drawing here. We've got some mixed micelles, but we've got some free bile acids. So we've got an in-between situation. And the truth is that in this situation, the injury to the bile ducts are nothing like as severe as they were in the original patients who've got no phospholipid in the bile. And I believe that the relationship between phenotype and the genotype or protein function for MDR3 doesn't look like a normal recessive curve and is much more like this. So the original patients are at the bottom left-hand corner still, but now we've got patients who are heterozygotes in the middle who don't have that phenotype, but they have a more slowly progressive cholangiopathy that may take decades to occur. And indeed, we've got patients who've got partial function on both copies who are indistinguishable from patients who've got one completely normal allele or one completely loss of function allele. And they both end up in the middle of that graph where they've got slowly progressive disease, sometimes taking decades before it clinically manifests. So this was originally described as a recessive, and we all thought that the patients will be, either at the bottom left-hand corner or fine. But now we realize this is much more complicated, and this has huge implications, of course, for genetics, because it now means that when we find a new child, we then have to investigate the parents and see, well, not only are they heterozygous so-called carriers, but actually heterozygotes manifesting disease. And so they require not just genetic intervention, but they also require some investigation to see whether they have got liver disease. Equally, if we've got adults presenting, then we need to start looking at first-degree relatives of them to see whether they have in fact got cryptogenic disease that's not manifest. The good news is that the milder forms of MDR3 deficiency actually respond very well to oral ursodeoxycholic acid, which is a more hydrophilic, much less detergent bile acid than the ones we make. And it's been shown that even patients with biallelic disease, if they've got missense changes, they're much more likely to respond than if they've got homozygous nonsense changes, for instance. And the data are so good, particularly generated by my French friends, that now ursodeoxycholic acid is specifically licensed in France for the treatment of MDR3 deficiency. And certainly the data so far are entirely consistent with these later onset mild variants being responsive to ursodeoxycholic acid. The problem we have is that they tend to present very late in the disease. So these are really worth identifying because if we intervene, then we have a very good chance that we're going to stop them getting progressive liver disease and all the complications thereof. So I think it makes it obviously much more muddy, the fact that we've got this very variable presentation in adulthood. But I do think clinically this is now quite a bit of extra work, but I think it's highly worthwhile. So if we contrast those two genes, BSEP encoded by ABCB11, there does appear to be a threshold. It basically behaves like a recessive, but we've got this common variant, which means that particularly if it's in trans with a damaging variant, we've got these patients who are really hovering on that 25% threshold and are predisposed to cholestatic episodes. By complete contrast, we've got these patients with MDR3 deficiency caused by encoded by ABCB4. There doesn't seem to be a threshold. There's a much more complex relationship between amount of protein function and the likelihood of having a phenotype. Okay, we will talk about the third different gene. That's going to be the last one I'm going to talk about in detail today. What we're looking at here is a rather beautiful electron micrograph showing 5 bile duct cells, cholangiocytes, and at the top, and it's the case in all epithelia, sealing the two adjacent cells are tight junction complexes shown by this electron dense material, which is the proteins making the tight junctions. And TJP2, which I told you earlier, and I'm showing again here, otherwise known as ZO2 or zona occludens 2, is actually a cytoplasmic protein which is closely integrated with the tight junctions themselves, but isn't actually the tight junction proteins which cross the membrane. Now, we can see on immunohistochemistry, we can see it at the surface of the cholangiocytes between adjacent cells with brown staining on the top picture and in decorating the canaliculus, which we talked about earlier, which is the space between adjacent hepatocytes at the bottom there. And we have shown using, actually this was two groups, there's a group, our group in London and a group of friends in Cincinnati, and we basically came up with the same results, so we combined the data and published them together. We used iPSC-derived hepatocyte-like cells, either patient cells or CRISPR-Cas mutated cells. And we first of all showed that if you take out TJP2, there is no paracellular leak. So in other words, the absence of ZO2 or TJP2 doesn't disrupt the tight junctions in terms of sealing the space between adjacent cells. And that's what's shown in the middle there, that there is retention of the barrier function, and different weights of dextran did not cross these tight junctions.
I'm sorry.
However, if we then grow these cells up, you can see at the left-hand side, you can see green BSEP en face on the top picture, but then on the slice on the second panel, you can see that green BSEP is in the apical membrane where it should be. But if you look at the two pictures below, one of which is a knockout TJP2 and one of which is a patient-derived cells, you can see that there's some BSEP on the surface, in the bottom one particularly, but there's also retention, intracellular retention. And if you look at these pictures, which is using the same sort of cells, but now we're looking at different proteins, and here we're highlighting in green NTCP, which is a basolateral protein. It shows up basolaterally in these normal cells, but is now appearing on the apex of these mutated cells. So what it looks like is, in the absence of TJP2, even though the barrier function is intact, we've got a problem with polarization of the cells, and we've got proteins ending up in the wrong place, so that the protein sorting is affected. Now, TJP2 was actually originally described in the Amish, as have several liver diseases being first described in them. And in the Amish, there is a single missense change, which is valine 48 alanine, which causes early onset cholestasis, which seems to really essentially entirely resolve by 5 years of age. It's not fully penetrant. Not everyone who's homozygous for that develops cholestasis at all. And Laura Bull, who's my long-standing collaborator in San Francisco, and I are putting a paper together which includes, I think, 95 patients from the Amish community who are all homozygous for this change. It is significant cholestasis, and in fact, the biggest significance is that they can present with late onset vitamin K deficiency because of cholestasis, and 4 of them died as a consequence of bleeding. But it is an early onset disease and associated exclusively with this one missense mutation that seems to get better. What we published just over 10 years ago was patients with biallelic truncating mutations who got early onset severe liver disease, do not seem to have responded to partial external biliary diversion, and the vast majority have been transplanted. So this is the other extreme, it seems, with 2 protein truncating mutations and a severe unresponsive disease. What we've now got is the next group of patients who've got early onset disease, a little bit later than the original ones, but these are patients who've got they've got biallelic variants, but they've got at least one missense change. So again, a very simple classification. But here the majority have remained transplant-free to date, and the majority who've had the partial external biliary diversion to reduce their bile salts have remained untransplanted as well. So quite a distinct phenotype-genotype correlation would appear. Well, then we've got another missense mutation, which is exclusively seen so far in patients of South Asian extraction. These patients are all homozygous for this missense mutation here, this histidine 788 leucine, which is obviously a different missense mutation. These do not present, well, only one of the 8 patients I've seen so far presents with neonatal cholestasis. All the rest have presented with episodic cholestasis, either later in the first decade or in the second decade. And again, not all the people carrying these variants are penetrant. And we discovered this because, to my huge surprise, when we tested parents, we found one mother and one father so far who are homozygous for the same change. And in one case of the mother, she's had 3 pregnancies, she's never had any problems in the pregnancies. I've repeated her blood test numerous times, and she's never shown any sign deviation from normality, despite being homozygous for this variant. So we've got, I would say, 4 different phenotypes now associated with TJP2 deficiency and reasonable phenotype-genotype correlation. I'm not going to talk about all these other types of mechanisms of cholestasis, which I'm highlighting on the left-hand side, and the genes which are relevant on the right-hand side. The list of genes involved in cholestasis does get bigger and bigger every year. OMIM insists on calling them PFIC 1, 2, 3, 4, 5, 6, 7, which I think is the most confusing thing, not only because I can't remember which one's which, but also because actually they are spectra, as I've tried to show you. They're not all just severe early onset disease. And I think talking about BSEP deficiency or MDR3 deficiency, each of which are spectra, is a more useful way of talking about these genes and the phenotypes associated with them. So very quickly, I just want to end by talking about testing. And I think these are actually just adult data looking at the adult uptake of cholestasis testing by our laboratory, which, as many of you will realize, is in the Southeast GLH area, but we cover the Midlands and the Southwest. Patients from Cambridge send it up to the Yorkshire territory. The vast majority of testing is obviously, not surprisingly, from our own adult liver disease service at the bottom, but you can see that the Royal Free and Birmingham are the other main contributors of testing. Now, what we've just looking at the analysis which Sammy did for me, in pediatrics, we've got basically something near a diagnosis entirely consistent with referral genetics in about 12%, but we've got variants which we think are probably responsible for at least part of the phenotype in another 17%, so overall about 30% of our pediatric patients. Now, the way we're using genetics in pediatrics is at a fairly early stage of the diagnostic odyssey. We're doing it when we establish generally that they don't need surgical intervention to look for biliary atresia, and then they have genetics done. And by doing this, we're saving a huge amount of phenotypic investigation, basically biochemical investigation, which we used to do previously. And we now only use those biochemical tests to corroborate the changes that we found on the genetics. So we're using it in an early, relatively non-discriminatory way. However, on the adult side, which is about 40% of activity now, we are having an almost identical pickup rate in terms of really a complete explanation in a slightly higher percentage, and at least partial explanation in a very similar. So my adult colleagues who are sending these samples are using them in quite a different way, of course, because in adult liver disease, there are common causes that everyone thinks of, like alcohol, but there's also immune diseases like primary biliary cirrhosis, primary sclerosing cholangitis, and all the viral hepatitides, and nowadays the metabolic associated steatotic liver diseases. But when the common diseases have been excluded, or if patients have not responded in the normal way to the treatment for those more common diseases, they are sending genetics, and we are now finding a diagnostic changes, or certainly clinically useful changes, in much the same proportion of those tested that we find in adults. In pediatrics, we've got hits in a whole range of genes, and ABCB11 is BSEP there, and MDR3 is ABCB4, but a whole range of rarer diseases, and obviously pick up in those genes that I'm showing you there. On the adult side, it is quite different. It is completely dominated by MDR3, encoded by ABCB4, which is the phospholipid transporter abnormality. The panel of genes where we're finding variants is much more restricted in the adults, and we are actually contemplating producing a smaller panel for adult investigation, as it will speed up analysis if we don't include quite so many genes. So I think this is where we are today. I hope that we're not thinking in terms of making diagnoses anymore. What we can do is we can use genetics to unravel normal biology and the pathobiology of these patients. We can identify variants which are contributing to patients' phenotype, but to do that properly, we all need to understand what the normal biology is in order to then understand what the clinical relevance of those variants is to our patients. Thank you. Again, I just want to highlight the huge number of people that have pulled together the sort of information which I'm presenting you today. And I'm very naughtily going to leave the lab email address down at the bottom there in case anyone wants to get in touch about clinical questions or testing. Thank you very much. And I believe we now welcome questions which can be put in the chat, if I'm right, Francis.
Absolutely. Yes, I should have said that at the very beginning. But thank you very much, Richard. And if anybody has got any questions, please do get them into the chat. I was just going to point out also that we have many patient stories on our website, but one of those is particularly pertinent to Richard's talk today, because Daisy, who is the mum of Ralph, Ralph has PFIC2, though you probably don't like that terminology, Richard, but we've used that
BSEP deficiency, please.
There you go, BSEP. We'll have to change the patient story, Nikki. But anyway, her story is there on our website, so you can go and read from the patient perspective a little bit about how he presented, and I think there's really interesting story there about the effect on the child themselves.
Sure. I mean, OMIM does a fantastic job of cataloguing human genetic disease, but they do insist on coming up with these sort of titles of FIC1, etc., and I don't quite understand their obsession for sort of giving numerical titles rather than what I believe are more useful deficiencies of proteins. But I've tried to suggest that they might stop doing that, but they have resisted my arguments so far.
Oh, well done. Because my little bugbear is that I think we should be getting away from naming syndromes after people.
Oh, syndromes, names, eponyms are even worse. Yeah, because they have
Exactly.
no relationship to the pathophysiology at all.
Exactly. I think they're really unhelpful, and we should be, being a geneticist, I'm thinking about, at least let's think about either the protein or the gene itself, depending
Yeah,
on what the
absolutely.
mechanism of the disease is.
Eponyms have one useful function in life. It's just catching out medical students in exams. That's not possible.
Exactly. But we seem to persist, and there are lots of new syndromes arriving. Anyway, Richard, I do have a couple of questions.
Fire away.
The first is that, and this is really helpful because I haven't actually really thought about the liver very hard since about 1992
Well,
when
I
we used to
didn't
share
touch
an
on
office.
I didn't touch on allergial syndrome, so I know
No,
you did a project on that many years ago. Yeah.
I did very many years ago. But I wanted to come back to, you mentioned cholestasis of pregnancy and how some of that is due to ABCB11 and that
Yeah.
particular variant. But I just wondered, in the context of cholestasis of pregnancy, as in the round, what
Yeah.
proportion is due to that variant or indeed to genetic factors?
It's still a minority, annoyingly.
Okay.
So we've still got work to do. Obviously MDR3 is actually also causes cholestasis of pregnancy. And that's actually a more complicated story, which we won't go into all the details today, but it is no question that I think actually getting cholestasis in pregnancy is an issue itself. Obviously, it's got issues for the mother in terms of pruritus. It's got hugely important issues we now realize in terms of fetal loss, particularly late stillborns. But also, I think it's actually a very important stress test because we can identify people who are actually at risk of developing liver disease later on. So I think actually being picked up as cholestasis of pregnancy, it could be sometimes a lifesaver because those are the patients we need to follow because they're actually at risk of developing chronic disease later.
And do you think we're under ascertaining genetic causes?
Yes,
Okay.
yes. At the moment, obviously, 1% of pregnancies are associated with cholestasis. And if we investigated all of those genetically, we would swamp the whole service. So we've developed criteria to restrict that to the ones with more severe disease. But eventually, I hope that we will investigate them all and we'll find out the true prevalence of these genetics. If you look in OMIM, no, not OMIM, I'm sorry, NOMAD. If you look in NOMAD at the frequency of MDR3 or ABCB4 variants in the population, if you look at the ones that have been published as disease causing, and please note that not everything that's been published as disease causing necessarily causes disease to the extent it was originally described. If you look at the ones that have been published as disease causing and you look at their frequency in different populations, alarmingly, you'll find that in the relatively small number of African ancestry individuals who are in NOMAD, you'll find 1 in 4 of them carry one of the published variants.
Wow.
Now, please don't take it away from here that 1 in 4 people of African ancestry have got MDR3 deficiency, because that I'm sure is an exaggeration. But there's 10 variants which contribute to that. And I don't think it's 1 in 4, of course, that really have disease. But I do believe that actually in some populations, particularly what looks like the nomad African population, which is almost certainly West African origin, do seem to have a particularly high prevalence of damaging variants in MDR3. And I think that was a big story that needs unraveling.
Very interesting. Thank you. We might give that a bit more thought, particularly given thinking about other populations, which we do so very badly.
So there's a question here about alkaline
is.
phosph and gamma GT.
Yes.
So yes, this is a big interesting topic because in adult hepatology, gamma GT and alkaline phosphatase are referred to as cholestatic enzymes. The truth is they're not. They are enzymes, they're measures of bile duct injury. They're not really measures of cholestasis. They're measures of bile duct injury, and they're very good ones. In pediatrics, we tend to ignore alkaline phosphatase because, of course, it goes up in growth in particular, and so it's a very unreliable marker. So we've, in pediatrics, focused much more on gamma GT, which is, again, like alkaline phosphatase, released from the biliary epithelium and reflects bile duct injury. But I do believe in adult practice, you can use either. Unfortunately, lots of hospitals don't even include gamma GT in routine liver function tests, which is extraordinary and needs sorting out. But alkaline phosphatase, of course, does come from other places apart from bile. That comes from bones as well, which sometimes confuses things. But I think if you've got somebody who's got elevated alkaline phosphatase and or gamma GT in adulthood, look for common things. Of course, the most common, if they're female and 40, then it's most likely to be PBC. And the most important thing is to look for anti-mitochondrial antibodies. But in patients presenting like this, if they haven't got anti-mitochondrial antibodies, if they haven't got IBD suggestive of PSC, these are the patients who should definitely have genetics done. So they stand out as unusual from the typical presentation of adult patients with elevated biliary epithelial enzymes, alkaline phosphatase and gamma GT, but they haven't got PSC or they haven't got IBD or anti-mitochondrial antibodies. These are definitely the patients who should be investigated, and you have a high pickup rate. And they're treatable.
Thank you. I have one more question. And then if other people have other questions, please do put them in the chat. My question was about treatment, which is obviously fantastic that you're now able to treat children with BSEP deficiency pharmacologically as well as surgically. I just wondered, first of all, you talked about the siblings. So at what point do you need to start treatment in order that you can therefore prevent progression? And the second thing is, where are you in terms of, funding, nice guidance, health economics, all that sort of stuff that gets so complicated?
We've got these two drugs, and they both have the same mechanism of action. They both block ASBT in the intestine, and they were developed originally as anti-hyperlipidemics a long time ago. They do cause a bit of diarrhea, particularly in people who haven't got cholestasis, although they're reasonably effective anti-hyperlipidemics. But obviously statins took over and these were put on the shelf, but they've been resuscitated for this purpose. They're both completed pivotal phase 3 studies, which I showed the publications. And at the moment, only Odivix, about one of them, is licensed in the UK for PFIC and is fully NICE approved and prescribable in the NHS. Obviously, so far, we're really restricting it to people with very significant pruritus and progressive disease. They are very expensive. The exact details of the NHS discount are not known to me, but they are considerably less than they're paying in America, for instance, which in an adult dosing is the thick end of $1 million a year, which for a drug for life is extraordinary. However, we're not paying that much, but obviously we are being conservative about where we use them. But if someone, I will still, if I've got a new presenting patient, whether it's a sibling or a proband, we will still start ursodeoxycholic acid. We'll start something simple and cheap like rifampicin. And really, if those are the two that really impact generally, if they don't work and we've got a diagnosis, then we will be starting Odivix at the moment because that's the only one that's licensed. Obviously, different parts of the world, certainly some countries, they then have restrictions on they've got to show efficacy to continue drug after 6 months. And I think some of the decisions that have been made are a little bit arbitrary, but we will obviously move on to other therapies, whether it's surgically or full-blown transplantation if we've got lack of efficacy, because that's really the only options we have at the moment. Obviously, we've got small molecules coming down the line which will increase expression of these proteins, which I believe are going to have an impact very much like the CF molecules. And further down the line was definitely people working on gene therapy solutions for these diseases specifically. Not because there's vast numbers of these patients, but actually they make good models because obviously we've got good natural history data, we've got good monitoring data, and we can really show whether treatments work. So I think the landscape is changing very rapidly, but at the moment we will start simple, cheap therapies early on, but then we'll progress quite quickly, usually in the first year. The drug's only actually licensed from 6 months of age anyway, so we will almost always have to start something else first. seem I haven't talked about Alagille very much at all. Just to go back to that, we've also got pivotal studies with both drugs showing that they are effective in Alagille. And we've also shown that if you respond biochemically and physiologically, we've shown that they increase transplant-free survival at least out to 6 years so far. We've published that. At the moment, maralixibat is the only one that's licensed in the UK for the treatment of Alagille syndrome, but is still not NICE approved. So we cannot prescribe that yet on the NHS. So we've got various early access patients, but we can't routinely prescribe it for Alagille syndrome, unfortunately.
Okay, thank you. There are a couple more questions. I'll go with Jens Yu Meow, which you can probably read, but EASL guideline for adult GC advice.
Genetic cholestasis.
Genetic cholestasis there for me, it's genetic counsellor.
Of course.
Yeah, yearly follow-up for individuals with monoallelic, biallelic variant receive yearly follow-up and ultrasound based on the phenotype. And if they have abnormal biochemistries or not responding to or whatever, do you agree with the guidance? What's your follow-up protocol?
Well, that's an easy one, really. Seeing I'm an author of those guidelines, I obviously agree with them. But not everything there is as good evidence as everything else. So if you read the guidelines, do look at the evidence base for everything. And those guidelines were written 18 months ago. Things will change slowly over time. But at the moment, I still think that's correct, yes.
And there was a question in the Q&A about alpha-1 antitrypsin deficiency, and I think this is referring to the surgical diversion.
We have no evidence that that's of any benefit whatsoever, and it would not be recommending that. I do think retention of bile acids is an important part of the problem in alpha antitrypsin deficiency, and I think we would not be recommending that or the use of these drugs in alpha-1. I'm pleased to say for alpha-1 there's all sorts of industry-based drugs coming down the line to reduce expression of the mutant protein and increase expression of normal protein by various different mechanisms. So I think alpha-1 is again a very variable phenotype, but a relatively prevalent disease. So there's a lot of industry activity in that area.
Great. Thank you. I think we're going to wrap up now. I'd like to just say thank you very much, Richard. Richard is coming back on, I believe March the 26th to give a slightly different talk, but along the theme of examining recessive and dominant inheritance. And we have a whole meeting which we're calling Beyond Mendel and challenging the assumptions of sort of traditional Mendelian genetics. So look at our website for details of that as well.
I've obviously hinted at some of that today.
Exactly.
So we will expand on that.
He will expand on that. And we have 2 other speakers talking about other aspects of that as well, that subject as well. But I'd like to thank Richard very much. Please do have a look at our website. Do join us for our other things today. Thanks, everybody.